Yep. So in this lecture, I will talk about human motor systems. And I will cover some aspects, including the motor behavior in humans and also neurobiology of the human motor system, as well as some control principles. So as we have learned robotic control in previous lectures, maybe you have some roughly idea of the motor planning and even some control manipulation of robots. So if assuming you want to design a robot to perform kind of manipulation task, so what it would be look like. So here is an example. In this video, we see this is a kind of a modern robot which can perform this manipulation task. And this is actually kind of a PhD project, and it's actually quite impressive. The robot can open the cover of the bottle and later pour the water and do a lot of quite interesting tasks as we do in the daily life. But in robotic control, there's a problem that such kind of task cannot be generalized. That means if you want a robot to do another manipulation task, you need this is another PhD project. So it cannot generalize. Another thing we also notice here is that the movement of the robot is not so, it's kind of clumsy. So if you compare to our human motion. So you see it's kind of very slow, the movement is very slow and also kind of discrete, not so smooth and continuous. So, but even though this is impressive and this is also like one of the state-of-the-art robots. And next example, I want to show you a human subject. So let's see what this little kid can do. And this child can do this very complex task within 5 seconds. And I want to understand what's happening in his brain and also in his motor system. And that's why we got interested to study human motor control. And then we can have a look at a comparison. Let's make a comparison between the robot we see and the human. You see a lot of difference actually. So start from the force execution. So robot is the force is produced by the torque motor. You know this if you're from the engineering background. So this kind of torque motor is very powerful and usually is linear. That means if you give the input, it immediately produces the output or the force that you require. But in comparison, in humans, you have these muscles are soft, which means when you innervate a human muscle, it doesn't immediately produce force. And if you look at the curve, it's actually a higher linear exponential curve. We will look at this muscle characteristic later on in the lecture. So you see the difference of this actuator of force production. Also, in robots, everything is information is transmitted by the electrical wires, which have practically zero delay. And in humans, if you consider this neural transformation, for example, for the arm movement, you need at least 20 milliseconds for the signal from the periphery to your central nervous system. And this is actually overall in the human motor system. And if you ask the roboticians to design a robot with such kind of delays, this is definitely not possible. Also, robot has very modern, accurate sensors. You can measure a lot of variables that you want. And your humans, these sensors, we will also look at these sensors later on. These sensors are not so precise and not reliable. You cannot really rely on the data that the sensor goes by because it also have a lot of noise during the neural transmission. So despite of this problem, it seems like it's not so advantageable for human with such kind of equipment. But still, it's clear that human can perform much better than the robot. So in this lecture, I will give you an overview of the human motor system. So this is a picture including all the major motor apparatus and the nervous system for motor control. So we have the brain, of course. This is probably the most important part. However, maybe less let's know is the spinal cord. This is also very important. We will come to that later. And the brain and spinal cord are called central nervous system. And also, of course, the muscle is important. That is what we produce force. And the muscles is innervated by the spinal cord. We see all these loops. So like there are loops going from the brain down to the spinal cord and to the muscle. And also there are sensory feedback loops from the muscle to the spinal cord and also all the way going up to the brain. So we will look at this in the following lectures. So this lecture will look at three aspects. First, we look at the behavior level. So we look at the movement. So what is the kinematics? And I will show you that when we look at human movement, we see some regularities in movement patterns. So it's not you cannot produce arbitrary random movement. So and then the second part is muscles is important force executor. So, and muscles are innervated by motor neurons, and motor neuron also coupled with reflex loops in the spinal cord. And we will look at different neural circuitries in the spinal cord. And in the last part, of course, the most important, the holy grail of the movement neuroscience. So we want to understand the brain. Of course, we are still not so clear about the brain function, but in this lecture, we will learn something about the basic anatomy of the brain, and hopefully I could provide some information on the function of these brain areas. And okay, so let's start from the movement. So I'm going to talk about this kinematic regularity. So this is a very simple example. If you do a pointing movement with a pen, so start from different starting point to the target flag, and you can move with different speed. So, and then what we found is that if you move faster, then the accuracy will be less, and slower you have become more accurate. So this is kind of intuitive. It's not so surprising. And if we plot this x-axis is the movement speed, and the y-axis is the endpoint error, and we see this more or less linear relationship between those two. And then we could also even make it more mathematical stuff out of it. So if we can make a model of that. So this is a more advanced version of this experiment is Fitts law. It's a kind of speed accuracy trade-off. So this is a modified experiment. So we can introduce a target width. So this W means that if your target is wider, then it's definitely easy to reach. And also there's a distance A in this term. So this term is called index of difficulty. That means if this term is larger, which means you have longer distance and more narrow target, it's more difficult. And if we plot the relationship, so of course we have movement duration here with this equation. And if you plot this x-axis as the index of difficulty and y-axis is movement duration, we can see a curve that could be explained by this mathematical model. But of course, this model doesn't tell us about any mechanism of this movement. It's just a kind of description and curve fitting. So another example is when we make a circular movement, so like drawing. So here we look at the velocity and curvature. So curvature is 1 divided by the radius. If you go to a circle, so that is if you have a more straight, more flat straight path, the radius is larger, so curvature is low. And what you see at this lower curvature, you have faster velocity. And when you go to this narrower curve, your movement is getting slow. So you can plot this. So if you plot the curvature and movement speed, you see also this kind of course, this is, you need some exponential function of that. So you can still get a linear relationship. So this shows some kinds of regularity when you produce movement. And let's make a more practical example. If you draw a letter A, so you see at this part, like point 2, you have more, this is curvature is less, so you move faster at this point. And when you go around this curve, then the velocity becomes slow. And so you can also explain the velocity at other locations. Okay, so the next example is when you do a reaching movement. So this is a horizontal movement. So you are the participant is sitting in a chair and they put the arm on the table. So this is usually a robotic device that you grab the handle and you can move different locations on the table. And we plot the kinematics of the hand and the joint. What we see here, when you move at different targets, between different targets, the hand speed is kind of invariant with this bell-shaped velocity profile. But if we look at joint angle, it can be complex. Its joint angle depends on the target, target direction, but not the hand speed. So this shows that the regularity is really in the hand space, but not in the joints. And let's dig a little bit further into this scheme. So, or not yet, but here, this is also if we vary the distance, and we look at different velocity move. So you start from to different distance. And we can see it's also independent of the target size or target distance. It's always, the hand is always bell-shaped. Acceleration, because this is just a derivative of this curve. So there's a biphasic profile of acceleration. So this is also regularity independent of other factors. And if we look a little bit deeper in the math to concretely model this, so then we can we introduce this minimum jerk model. So jerk is the derivative of the acceleration. So here you have here this x is the position, and then you have the velocity. Then derivative, you got the acceleration, which is biphasic, and then you get another derivative. It's called jerk. And if we define a cost function as the square of the jerk, and then we use optimization method to get the minimum cost based on this function, then mathematically we can get the solution of this x traces, which is this. And the more detailed mathematics is you can look at in this material, but I don't want to go to in details. So the motivation of doing this is that because by minimizing jerk, you can have produce this very smooth trajectory. So that is this is still a kind of curve fitting, but it's good like describe some features that invariance where you produce movement kinematics. Okay, and let's see another example here. So when you move in a perturbation, with a perturbation, so here still you are doing this horizontal movement, but you can here, for example, there's null field, means there's no external perturbation. So you see the participant is moving, they start from the center and they go to different targets and they always make straight lines. So this is not surprising. And then we introduce this perturbation. So this is a very complex force field. It's called a force field, which means the perturbation, the direction of the perturbation and the size of the perturbation depends on the hand velocity. And this can be shown here. So that is actually very complex. If you move, for example, if you move vertical direction, then the perturbation will drive your hand to the right side. So it's actually a very difficult task if you do this experiment. So that's what we see at the initially when we expose the participant to this force field, they make curved trajectory. So they cannot finish the task in one single movement. They always have some correction near the movement end. But eventually, when they can be trained, they keep practicing, and we see after a certain time they are able to make straight movement. This is called adaptation. So you see, if we compare the case in adaptation with the perturbation and the null field without perturbation, you see the subject still tend to make straight lines. So this is kind of intuitive invariant in when you produce movement. And interestingly, if you suddenly remove this force field and the subject's not aware of that, then the second trial, they will do this inverse error. So here, that is, this is also very well-studied task in human motor control research. Okay, so now we have look at several features of invariance in kinematics. So let's summarize a little bit. Here we start from the speed accuracy trade-off, which is Fitts law, and also velocity curvature relationship is called power law. And then we look at this planar movement. We see the bell-shaped hand velocity, and using minimum jerk model, we can describe these relationships. And we also look at the force adaptation, in which subjects always tend to make straight lines. Neurally, actually, I'm not included in this lecture, but there are also some neural data showing that in the brain, you also have, if you plot the neural activation in the time course, you also have similar features of capturing these kinematics, but I will not go to these neural lectures. Okay, so this is the first part of the motor behavior. So let's come to the muscle. Muscle is the only executor of movement in humans. That's why it's very important, as highlighted here by the Nobel Prize winner, Charles Sheridan. And here we want to understand or want to learn how the muscle look like and how this is controlled. So if we took a muscle here, this is a biceps on the upper arm. And we look at more deeper into the structure of the muscle. We see that the muscle consists of many muscle fibers, these small tubes. And each muscle fiber is innervated by the axon of motor neuron. And the motor neuron is a type of neuron in the central nervous system. And assume that you all know what a neuron is. And the neuron is consisted of the cell body and the dendrites, which receives sensory input, and the axon that send out the signals. And this is an electrical chemical device that's transferred informations. So, if we took one alpha motor neuron, it can innervate multiple muscle fibers, and together with all these muscle fibers, this consists of the motor unit. So motor unit is defined by the alpha motor neuron and the muscle fibers that it innervates. And because the muscle consists of many muscle fibers, so you may need many alpha motor neurons for control of one single muscle. And these alpha motor neurons forms a group which is called the motor neuron pool. And let's see what is the force production in the muscle. So here you have, we can record the neural activity of a muscle. So, so when you give the input to the muscle, this innervation is you have a smaller input, for example, a single action potential. What you see in the muscle response is a twitch. So like a peak in the force that muscle produces. And when you increase the frequency of the stimulus, and you see these peaks add together, and then if you reach a very high frequency, the peaks becomes less clear, and then it forms a smooth curve of force. So that is also basically this is how our muscle system is producing force. So with higher frequency stimulation. And muscle is, as we see, muscle is innervated by motor neurons, and these motor neurons also embedded in the spinal cord. There are motor and sensory pathways. So first of all, let's look at the spinal cord. So spinal cord, in the image, if you take an image of spinal cord slides, usually we distinguish that between gray matter and white matter. So gray matter looks in the image, it looks like gray color. And this is mainly the cell bodies of the neuron located in the green matter. And in white matter, it's basically these axons. So, but this is just this butterfly shape is just an illustration of this. You can roughly divide this spinal cord neurons into these different distinct sections. And the alpha motor neuron, so is the cell body of the alpha motor neuron is located in the ventral root. So the ventral root means in the front, ventral root of the spinal cord. So here's the cell body of the alpha motor neuron, and then it goes through the ventral root. It goes to the muscle where it innervates muscle fibers. And in the other direction, so from the muscle to the spinal cord, which is the sensory. So if you have a sensory receptor on your hand, and then it receives sensory information and it goes back through this called dorsal root ganglion and going back to the spinal cord, which terminates on the dorsal part of the spinal cord, which is on the back. And the cell body of these sensory neurons are located in this, actually outside of the spinal cord, which is here, the dorsal root ganglion cell. Okay, so this is basically the structure of this input-output pathways in the spinal cord. And if we look at the inside the muscle, so we have this is a muscle fibers that produce force. But if we dig into these muscle fibers, we also see some smaller structures. This is called like spindle-like structures. This is called a muscle spindle. And what are these things do? So muscle spindle is a sensory organ that you see there are smaller muscle fibers here, and there are sensory axons that are wrapping around these thinner muscle spindles, and they goes back to the spinal cord. So when your muscle is stretched or change length, these axons will sense the change of muscle length and give the information back to the nervous system. So that's we say that the muscle spindle is a sensor of muscle length. And this is called the muscle spindle. The fibers as muscle spindle is called the intrafusal muscle fibers, and the muscle fibers that produce force is called extrafusal muscle fibers, which is summarized here. So extrafusal muscle fiber produce force, while the intrafusal muscle doesn't produce force, but only sense the length of the muscle. And as the alpha motor neuron innervate extrafusal muscle fibers, there are also another type of neuron called the gamma motor neuron that innervates the intrafusal muscle fibers. So as we see here, so this motor neuron terminate on the end of this ending of the intrafusal muscle fibers. And then you may ask questions, so what's this for? So why I want to produce force, I just use alpha motor neuron to innervate these larger fast muscle fibers. So why should I still also send through the gamma motor neuron to innervate the intrafusal muscle fibers? So that's actually an interesting question, and I can explain you in the next figure here. So here we see what is the function of this gamma motor neuron. If we take an isolated muscle, here we plot for clarity, we plot the extrafusal muscle fiber on the left and the intrafusal muscle fiber or the bundle spindle on the right. So they are also the changes, they are parallel change the length. So if we attach a weight, then we expect that there's a tension in the muscle fibers, and because the muscle spindle is also same time lengthened together with the muscle. So you will also have the sensory information here when the muscle is stretched. Then we stimulate the muscle fiber by alpha motor neuron, so the muscle becomes shorter. At the same time, the muscle spindle becomes also shorter. And then it becomes loose. You see, this is not very tense anymore. So then you cannot give any information here. So that you will lose the information of the sensory signal once you stimulate the alpha motor neuron. But if you simultaneously use the gamma motor neuron to stimulate the muscle spindle, then the muscle spindle becomes tight again, even though the muscle is shortened. And in this case, it still gives out the information of the muscle length change. So you can see by this gamma motor neuron, this the activation of this sensory feedback is maintained. And by modulating the intensity of the gamma motor neuron, you can modulate the sensitivity of the muscle spindle. They're still not quite clear, so exactly what the function of this, why I should keep this information here. But researchers have done some interesting experiment, as we see here in this slides. So here, I just draw the picture differently, just reorganize the same as we have before. So you have alpha and gamma. So alpha innervate extrafusal muscle fibers, while the gamma innervate muscle spindle. And here, from the muscle spindle, you have the sensory feedback back to the alpha motor neuron. And then the cortical, the brain, while your brain innervate the spinal cord, it simultaneously sends signal to alpha and gamma. And by this gamma neural loop to the spindle and back to the alpha motor neuron, you see this is all plus. That means through this loop, the alpha motor neuron activation is enhanced by this additional sensory feedback. So there's evidence for this loop, which is shown in this experimental recording. So here recorded is the EMG, which is the electromyograph, which shows the activation of the muscle. And here you have, this is a finger muscle when you do some finger movement. So sensory fiber activity also recorded here. And this is the same. Here we cannot record in the gamma motor neuron because this neuron is very small in size, so experimentally it's very difficult. But here we can indirectly say that the gamma motor neuron is active because in this case the muscle is shortening. The muscle is shortening, and you have muscle activation, and you also have sensory activation, right? If you are not active gamma motor neurons, you will not expect the sensory feedback because muscle is shortening. So in this case, it's indirectly indicate that you need to activate gamma motor neuron to get the sensory activation. Okay. So here, I think we already know this. So there are different sources of as input to the alpha motor neuron. You have the input from the brain to the upper level, and you have the sensory feedback through the muscle spindles going to the alpha motor neuron. There are also many interneurons in the spinal cord, and this interconnect the different loops, and we will look at later. Okay, so this is I think I already more or less described this loop before. So this is called a stretch reflex loop. We have alpha motor neuron that innervate this muscle. And so in human joints, you always have this pairwise muscle. So this is called agonist and antagonist. Agonist means when it assists the movement, while antagonist is on the opposite of the movement, and it usually breaks the movement. And so here, for example, here once you active this, for example, you active this. This is called flexion, right? Flexor. So this muscle is flexor. If you innervate flexor, then flexor change length, and then you have the feedback loop of this through the sensory feedback, and because and then it's also so if your muscle is stretched, the feedback is increasing, and then going back to this muscle, it will increase the alpha motor neuron activation to make your muscle shorter. So that is a stretch reflex, which also always resists muscle length change. So this is kind of a negative feedback loop. And at the same time, when you activate this sensory feedback, it going through an inhibitory interneuron, it will inhibit the opposite side. So that means it's if this stretch reflex increase activity in the agonist muscle, it will simultaneously decrease activity in the antagonist muscle. So this is quite a kind of synergic cooperation between the two muscles. This is also the design is called a reciprocal inhibition. It's a design to make some function of this loop. There are also other mechanisms or loops in the spinal level. Here is the Golgi tendon organ reflex. Golgi tendon is the structure between the bone and the muscle structure. So here, and there are also sensory nerve that transmit tendon organ property back to the spinal cord, and then through an inhibitory interneuron going to the alpha motor neuron. So this is an inhibitory loop. That means if you produce muscle force, say if alpha motor neuron is active, muscle is shortening and then muscle force is increasing, then through this tendon reflex, it will inhibit the alpha motor neuron. So that will make your alpha motor neuron less active. And this is this kind of a resist of the increase of force. And this tendon reflex is thought to protect the muscle. So if your muscle have too much forces, and it helps to prevent damage of the muscle. There are also other more complex loops in the spinal cord. So here we already show that this inhibition. So here we already introduced before. So we have this reciprocal inhibition. And more complex is that these interneurons can also be modulated by the signals from the brain. So that means the stretch reflex loop can be modulated by the descending activations. And this here we have this rational cell. It's very interesting structure as we see here. So the rational cell is shown here in the gray color. It always couples to the motor neuron. For example, here this motor neuron, once it innervates muscle, it also innervates rational cells, and the rational cell in turn also inhibit the muscle, this alpha motor neuron activation. And so this loop is always like not let the motor neuron active. So this is always there. So, and still it's not very clear what exact function of the rational cell, because it's also modulated by the descending pathways. So depending on different tasks, you can have different input to this rational cell. And also it projects to the opposite side, to the antagonist. And it also have the opposite effect on the other side. So there are actually many loops in the spinal cord, and still the function of this loop, how they are integrated and what is the function is, many researchers are doing work on this, and still not quite clear that how everything is integrated together. So here is a research paper that doing the simulation. So basically they implement all these well-known structure of the spinal circuit. For example, this R is a rational cell, and we have 1A, this is a stretch reflex loop, and alpha motor neuron. 1B is the Golgi tendon reflex, and the PN is another, it's called the propriospinal neuron, which we haven't talked here. So anyway, they put basically all these spinal loop circuitry together in this model, and they want to simulate the movement. And very surprisingly, if you just give this go signal, this input to this circuitry, if you just give a step function, very simple input, you can see this muscle in simulation. They can produce some reasonable movement. So this simulation work tells us that to control movement, you don't need actually very complex signals of the brain, and a lot of things can be done automatically through this complex neural circuitry. Okay, and then let's come to more conceptual understanding of the muscle control. So how do we understand this? So we introduced the analogy of mass spring model to compare with the muscles. So we know this mass spring damping system. So we have the elastic component have reached the stiffness, and also the viscous component C and attached to the mass. So this is can be well described by a dynamical system. And for simplicity, let's first ignore this damping component. So let's just think about the muscle as an elastic spring here. So what is the property of the normal spring? So if you press a spring, this gets shorter, then it has some force to resist the push. And if you pull the spring, then the spring gets longer and you also have force to resist this length. And so muscle have a similar spring-like behavior, just like a normal spring, except that the muscle can only act in one direction. So you can only pull the muscle. If you push the muscle, you don't produce force, right? Because the muscle becomes sluggish. And this is one difference. So if we want to model the muscle, you can also use one side of this characteristic. And also because of we also have this agonist antagonist muscle pairs. That means the force is always two sides. That means even you can only pull one side, but if you pull the other side, that is equivalent that you push the other side. So you can adjust these two springs to make movement. So this is bidirectional. And also this so what results in this spring-like characteristic of the muscle? So we have both passive mechanics. So if it's nothing to do with antineural, so just like a passive. So mechanics, you have the elastic. Also, you have the reflex. As we see, you have the reflex loop. Once you stretch the muscle, it will inhibit. It will try to resist the lengthening. So this reflex mechanism also contribute to this elastic property of the muscle. And to illustrate this, I show you many experimental results of the muscle properties. Here you see all these kind of exponential curve that represents the property of the muscle. It's like a spring. So first of all, this curve. So that means if you stretch, you lengthening the muscle, the muscle force becomes larger. And this property is also as a result of both spinal reflex and the passive properties of the muscle. And what is interesting here is in the experiment, of course, in humans, sometimes it's difficult, but in animal experiment, so for example, here on the left side, the experimental don't want the animal to produce voluntary movement, so they remove the brain of more exactly, they remove the cortex of the cat. And then the cat will not have the will to move. So the cat is just not dead, but it cannot move, voluntarily do the movement. And then the experimental will stimulate the brain and to make artificial input to the system. So that for example, if they take strong stimulus, we will give a stronger stimulus, and we see the muscle will stay at a certain length. And then in this condition, the experimental stretch the muscle, and then the muscle have this kind of characteristic. And in the second time, the experimental make a less stronger stimulation to the brain. Then you see the muscle length or the zero length of the muscles have a different value. And at this length, you also stretch a muscle, you also have a similar curve, and so on. So you change the intensity of the stimulus, and you see the zero length of the muscle changes according to how strong you stimulate. But the general characteristic of this curve doesn't change, right? It's just a kind of a parallel shift of this. So this is done by in the cat leg muscle, soleus. And they also do some other muscle, this GS is gastrocnemius muscle, and by stimulation of the vestibular system, and we see the similar curve that also this the zero length change depending on the stimulation, while this shape of the curve doesn't change. And this zero length, let's call it lambda here for now. Let's use this notion. And in humans, we cannot do in the same experiments like in cat, but we can ask human subjects to do some task that say you don't do voluntary movement, but when I stretch your muscle, you don't resist, just let it go. So you can still kind of measure human muscles and by asking them not to intentionally resist. So you can still at different location or different lengths of the muscle or different joint angle, you can have the measurement of to plot this similar length force characteristic of the muscle. Also, even in the eye muscle, you can do that. In some surgery, the doctor also tested this. They also stretched the eye muscle, and surprisingly they got also similar curve of the eye muscle here. So how can I make use? Can we make use of these characters of the muscle property? Let's still consider the mass spring model. So here we have length and force. So the lambda is the zero length. So once your brain specify a lambda value, the zero length value, the curve is determined. Depending on the external condition. So if you have an external load, for example, you put some force into the muscle, then this force will have an intersection with this curve. This point is called equilibrium point, EP. That means if the system, that is the stable point that the system arrives. So in the dynamical system, so we'll have learned this dynamical system language, and we can consider the EP as an attractor under this condition. So if your system is not at this position, then it always goes to this attractor. So this is some description of this character. And then how can we make a movement? So if you want, what about if we want to move? So what you do is you change the zero length, which is this lambda here. So you change, for example, if you want to shorten the muscle, you move the lambda toward the left side, which shortens the muscle. This is an active movement that you change the zero length. At the same time, the curve will shift together with the lambda. And then this, with the same load, this curve and defines a new EP position, this EP2. The previous EP1 becomes a deviation from the EP2. So that means according to this scheme, so you have a new EP here. So then the system will be attracted to the new EP position. With this scheme, it's makes sense of how the muscle and muscle length and force are controlled, and by introducing this equilibrium idea. And for the passive movement, sorry.
Just question. Yeah. Force is the force that which the muscle is activated. It is not the force that is externally applied to it.
In this case, for example, if you are at this location, this is a stable point, right? Maybe that's stable point. In this condition, the force, the muscle force, and the external force the same. Okay. So this is both muscle force and external force. Because you cannot only have muscle force, right? You have at the stable position, if your muscle produce force, you have to somehow fix the muscle, right?
Okay.
So externally, you should have also some force applied to make the muscle stable, right? So that means the external force and the muscle force is the same in this picture.
Essentially on the entire curve, so the curve describes
where
the forces.
So this is the curve. So each point on this curve is an equilibrium position, equilibrium point, depending on the external. So if you don't have this force, the equilibrium will be here, right? The force, external force is 0, you will have this point. And if you have different external force, you will have different EP position. And in this case, you have this L as if your external force is L, then it defines this EP position. So depending on external force. So this y-axis is actually the static force where muscle force equals external force. Yeah. So it's actually both. Yeah. But it's difficult to think about dynamic. If you move, then it's only a static case in this curve. And in passive movement, so in passive movement, that means your brain doesn't want to change muscle length. So that means the zero length is fixed. So that means this you have this curve here. But passive movement, if you change the external force, so previously we have L1. So if you decrease the external force, then the new equilibrium position will be defined by the intersection between this line and this curve. So then your muscle will be here. That means you will shorten the muscle and while the force decrease. So this is just a kind of illustration to help understanding how the force length changes. Is kind of the property of the muscle. And so now we are still in the muscle domain. So let's think about joint. So one joint is controlled by two muscles, right? Two sides. You have the flexor and extensor. So flexor has a zero length, and extensor also has a zero length. And notice that here this is the angle and torque. So previously we have length and force. So in this case, this will be not the muscle length, but the joint angle position that corresponding to the muscle length. So if you have the moment arm, you can calculate it. So it's basically equivalent as muscle length. So, but let's say this is joint angle, and so you notice that the flexor and extensor should have different direction. So if you go to the left, the joint angle decrease. So in this case, your flexor becomes shorter and the extensor should be longer. So this, for example, at this joint angle, you have a smaller angle, also called a zero length for flexor, and a larger angle for the extensor. And so if you plot the force for the extensor, have to be opposite sign. So that's why this goes to the negative part. And if you sum these two characters together, so you will have roughly linear line, which is called the joint torque. So that if you sum up two muscles, you will have a joint torque acting at the angle. And if you want to do a joint movement, so you have to shift both flexor and muscle zero length to a new position, right? To a new angle. Then this whole characteristic shift together to the new joint angle. And here we don't have external torque, so that's why the equilibrium position is also always at this with the intersection of zero torque. Okay. Yeah, so this is maybe not so easy to at the first time, but it provides you some insights of how this like mechanically also is introducing this equilibrium idea, how this the joint is controlled. And here is another case that if you what about if you just stay in the same angle, but only make this lines this stiffness stronger. So that means the slope becomes larger. in this case, you can see you have to make this this line, you have to increase, you have to change the lambda for flexor and extensor to the opposite side. So you have to switch the flexor zero length to left and the extensor to the right, such that the sum of them will give you this curve. And what is unique about this case is that the angle doesn't change, but the two muscle zero length change. So that will be this is called co-contraction. That means if you stiff your joint is not changing, but your two muscles produce force. So this explains this behavior, right? Yeah. Okay. So based on this, we can do some simulation in research. So first we have this so-called heel tap, not reading here, the heel tap muscle. So in this, we are modeling the muscle properties. So here, this C is the contractile element, which is the active muscle force. And also because you have, besides muscle fiber, you also have this muscle fascia and tendon. So this corresponding to the P parallel element and the serial element. So this whole model explains well the muscle mechanical properties and has been used a lot in the research of modeling of human biomechanics. And there's a software called OpenSim. They have very detailed and realistic biomechanical model of the muscles and joints. And currently in the lab, we want to use the heel tap model together with the stretch reflex model to build a complete, more or less complete circuitry for movement generation. Because we don't know what is happening in the brain, but if we have the model and we can measure the model output, which is the kinematics or muscle activation, we can inverse this model to estimate the model input, which is the brain descending signal. And this is the research work that we are currently working in the lab. Okay, so I think for the muscle part, we have learned about the basic anatomy and the structure of the muscle spindles. And also we see there are many stretch reflex loops. And also we learned about this nonlinear muscle model as a mass spring model. So I think in the third part of the lecture, we'll come to the most challenging part, which is the brain. So here I will talk about three major brain area which are responsible for movement generation. So first is motor cortex, which is the surface of this folding of the brain, and also cerebellum, which is located here, and also basal ganglia is located inside of the brain. We will look at individually. Of course, there are other brain areas like thalamus is also important, and the brainstem, but we are not going to this. So let's start from motor cortex. So motor cortex send their axons down to the spinal cord through a structure called cortical spinal tract, and we see here it terminates the axons of cortical neurons can terminate directly on the motor neurons, or they can go through some inhibitory interneurons and then control the motor neural activations. And in this picture, it's a slide of the spinal cord, and you can see the different descending tracks going to the spinal cord. But I think we'll not go into the detail of this anatomy here. About motor cortex, we can still divide motor cortex to several regions. So the most famous one probably is the primary motor cortex. That is called M1, which is here, this area. I'm not sure if you are familiar with this so-called homunculus. And this is basically a mapping from the M1 to muscles. So this, if you stimulate a point in the M1 area, you will see some twitch in certain muscles. You can measure that and you can map that. Actually, this experiment is done by a Canadian surgeon called Weld Penfield, like 100 years ago, when he did some brain operation because the patient scalp is open. So he want to check that the place they operate doesn't affect motor function. So he has to make sure that it doesn't touch the motor area. So he will test a different point of the M1 and to see, okay, if I stimulate this point, you have some movement in finger or in some arm muscle, and make sure, okay, I should not touch this point. And in this surgery, he tested different locations of the M1 area. And say, for example, if he stimulate here, then he see, okay, the hand muscle twitch. And then if he stimulate here, they see the muscle, the shoulder muscle twitch. And in this way, he can map different body segments to the M1 area. And this is a very classical finding of this neurophysiologist. It's called homunculus. However, this view is challenged by the recent research because people think that many research think that it's maybe not so simple like one-to-one mapping. Actually, if you stimulate one part of the M1, you can see response in many muscles. So it's not just not one-to-one mapping. Also, for one muscle, you can also have a neighboring area also contribute to the muscle. So it's not like that simple. So people still there are some recent paper that have some hypotheses about how this M1 is organized according to the lower level muscle. So, but this is just some roughly idea of how this distributed in the M1 related to the muscle. And there's another part of the motor cortex called the premotor area. Usually it's thought to be more responsible for motor preparation, but it also contribute to the movement generation. Here they do an experiment in monkeys. So they record the PMA neurons. We see that before the movement, so when the lights turn on, the monkey should reach. Before the movement, the monkey, when the stimulus is on, even without any movement, these neurons in the PMA area already fires. But when the monkey starts to move, the firing will stop for this PMA neurons. So it seems that for this neuron, it's kind of related with movement preparation, but not related to the movement itself. There is another interesting type of neuron. It's called mirror neuron in the PMA area. That is when the monkey doing a movement, the neuron fires. And when the monkey observe another monkey doing the same movement while it doesn't do the movement, the neuron fires too. And also even when he observe a human doing the same task, the neuron fires. But if there's no movement or some other type of movement, the neuron doesn't fire. So this is also interesting. It's called the mirror neuron. Okay, let's go to another structure of the brain, the cerebellum. Cerebellum is mainly responsible for the coordination of the movement. And let's have a look at the anatomy of the cerebellum. So it has you see it has interconnections with different brain structures. And I think maybe in the next slide, it's more clear. Yes. So if we take this cerebellum and look at its cortex, it's called cerebellar cortex. It's also kind of quite complex structure here. So it's mainly about different cell types. So you have here, so let's start from the mossy fiber. So here, this mossy fiber carries sensory information from the periphery to the cerebellum. And it connects with this granule cell and forms these parallel fibers. So the signal goes from here and come to the cortex and then connect with this Purkinje cell. So Purkinje cell is very large cell and easy to identify in the cerebellar cortex. So there are multiple cells. You see these multiple Purkinje cells. And then this Purkinje cell will send the information to the motor control area in the brain. And also there's a climbing fiber from other control area that couple together with the Purkinje cell. So here is basically the structure of this connection of different types of the cell in the cerebellum. And people want to understand what's the meaning of this connection. So what is related with movement control. So people have proposed several control models for the cerebellum. It's basically, we have two types. One is a forward model, which is a prediction of the movement. You can do some prediction. And the inverse model means that you can, given the output, by inverse model, you know the input. But this is very computational stuff, so we'll not go to detail of this control framework. But here they want to relate these control blocks to different parts of the cerebellum. For example, they consider the cortex, the cerebellar cortex, related with the inverse model. And for example, this climbing fiber, because this climbing fiber carries feedback information to this cerebellar cortex, so they match the climbing fiber as this error signal to this inverse model. And also they also have other like mapping different fiber types to the control scheme. Okay. This is just for you to know that there are some computational studies about this cerebellum function. And what happens if we have lesion in the cerebellum? So here we have some examples of the behavior of cerebellar patients. So first of all, if you ask cerebellar patients to do movement with both hands, so the lesion is on the left side. So cerebellum lesion left side will affect left side muscle. So you see the left hand will have a delay of the movement outside. Also this movement also not very smooth. And second example, if you do, this is also a very classical clinical test. So it's called a finger to nose test. So if you're pointing outward and then you go to your nose, normal people just do a straight line, straight movement. But for the cerebellum patients, they make large errors and the movement is not straight. And if you also ask the patient to do this flip the palm, and you see the normal people will do this rhythmic, but not in the patient. Another test in the cerebellar patients is this double joint movement. If you flex your elbow joint, normal people will not move your shoulder, right? So you can somehow compensate the interaction torque of when you move the elbow, right? For example, here, this is a net torque. That means your shoulder doesn't move. But in cerebellar patients, if you ask her to flex your elbow, he cannot make the shoulder stable. So the shoulder always moves together with the elbow. So that's why you see the net torque is not zero in the shoulder. Okay, so this is about cerebellum. Last part of the brain I want to talk today is basal ganglia. Basal ganglia is kind of, people think it's related with decision making and the initiation of the movement. So the structure is, it consists of several structures of the basal ganglia. It's located in the very deep in the center of the brain. And the most important part probably is this substantia nigra. This part is, I just want to talk a little bit about this part of basal ganglia, because this is related with the neurotransmitter, the release of the transmitter. And Bisogania is also associated with some reward. When you move, you want to get some reward, and this feedback rewarding mechanism is people think it's related with the basal ganglia loop. And this dopamine is very important because if you have dysfunction in dopamine production, you will result in disease that people discovered called Parkinson's disease. And in this disease, what you often see is this patient have difficult to initiate a movement. They have, in the static case, in the resting case, they have tremor, and they are freezing, they cannot move. But as long as they somehow manage, if he somehow managed to move, then the tremor disappear. And the cause has been identified as the loss of dopamine in the basal ganglia. But unfortunately, we don't know why this happens, why these neurons degenerate. Okay, I think I will show you a video of Parkinson patient. This is what we have described. So in the stable case, you have the tremor on the limbs. So and he cannot really make a step. So it can only someone help him to move. So it's kind of you are freezing. You cannot something stop you to move. That's a problem in basal ganglia and the dopamine. The problem with dopamine. Okay. Okay, maybe I can. In the second video is interesting phenomenon that if you ask the patient, the same patient, to ride a bicycle, you see he has no problem at all. It's just like a normal person of riding a bicycle, and he can even turn the bicycle going back. And you see when he moves, there's no tremor and there's no problem during the movement. So the problem is really initiating the movement. And as long as he steps out of the bicycle, he just freezing again, and I guess the tremor comes out. Okay, yes. Okay, so this is just some example if something goes wrong in the brain. Okay, so in the brain we have, I hope we have learned something about motor cortex, which is important for control of voluntary movement, and the cerebellum for coordination of the movement. Basal ganglia is also interconnect with many brain regions and important for movement initiation. Okay, I think so. Let's just do a short summary of this lecture. So first, we studied mimetic patterns, and we see human cannot produce random patterns. Instead, the movement patterns are always regular and follow some features. And we also look at how the muscle produce force and these neural networks in the spinal cord, the reflex loops. And then we also look at different brain areas and see what happens if something in the brain doesn't work, and about these movement disorders. These are the books from this lecture, and many of the figures I show here is from these books. Most of them are standard textbooks of neuroscience. Yeah, okay. I guess that's it.